Hello students, hope all of you are fine. Today we are going to discuss about some basic concepts of microbial genetics. Now, what is genetics? It can be simply defined as the manipulation of DNA to study cellular and organismal function. We all know that DNA encodes all of the information needed to make the cell and the complete organism. So the effects of changing this molecule can give clues to the normal functions of the cell and so the organisms. Before the discovery of methods for manipulating DNA in the test tube, the only genetic approaches available for studying cellular and organismal functions were those of classical genetics. In bacterial genetics, we usually deals with the genetic techniques that are used to study bacteria. One of the major advantages of bacteria for genetic study is that they are haploid. That means they have only one copy or allele of each gene. Another advantage of some bacteria for genetic studies is that they have very short generation time. The generation time is the length of time the organism takes to reach maturity and produce offspring. Some strains of bacteria of Escherichia coli can produce themselves every 20 minutes under ideal conditions. Another advantage of the bacteria is that they multiply asexually by cell division, means binary division from one cell to two cell. Sexual reproduction, in which individuals of the same species must mate with each other to give rise to progeny, which can complicate genetic experiments because the progeny are never identical to their parents. Let us discuss about the genome. The genome is the entire collection of gene or genetic material and all other functional and non-functional DNA sequences found in an organism. It includes structural and functional genes, regulatory genes, and non-functional nucleotide sequences also. The genome of all living beings represents their hereditary material and is formed of DNA. In simple prokaryotic cells, the genomic DNA forms a single circular chromosome. It is without basic proteins and lies in the cell cytoplasm in the nucleate region. In eukaryote cells, DNA is associated with basic proteins. However, some RNA is found associated with DNA and forms its backbone or core. Bacterial DNA is arranged in supercoiled loops. You can see here that these loops are affixed to the central nucleotide protein matrix and RNA stabilize the supercoiled state of DNA. In Escherichia coli, DNA is estimated to have 400 loops and each with an average length of 10 to 20 RBP. RBP means RNA binding protein. These supercoiled loops are dynamic structures and keep changing during cell growth and cell division. Some of the coils are supercoiled while others are relaxed. Supercoiling of DNA and also the DNA binding nucleoid proteins contribute to DNA compaction. These proteins are called structural maintenance of chromosomes proteins. In bacterial cell, RNA is not the integral part of the chromatin. The circular chromosome of Escherichia coli has a contour length of about 13,600 micrometer and it is about 20 angstrom wide. Its molecular weight is about 2.8 into 10 to the power 9 and it has about 50 or more highly twisted or superquint loops. It consists of about 4.6 into 10 to the power 6 nucleotide pairs organized into about 4,400 genes roughly. Now we will focus on the gene. Genes are the unit of inheritance. These are located in the chromosomes in linear fashion and are carried from parents to offspring. Genes control the expression of characters. The word gene was introduced by Johansen in 1909. According to the modern concept, a gene is a segment of DNA with specific nucleotide sequence which has a coded information for a specific character. The specific sequence of nitrogenous bases in a gene provides information for the synthesis of amino acids in the polypeptide chain during the synthesis of a particular protein. A gene is now described as a unit of function, a unit of mutation, and a unit of regulation. Now, depending on the functional activity, the genes are classified as follows. Number 1 is housekeeping gene or constitutive gene. The housekeeping genes are functional all the time throughout the life. These are needed for the basic cellular activities. Such genes are called housekeeping genes. For example, genes associated with encoding the enzymes for glycolysis are active in all cell types and all the time throughout their life. Their expression is not regulated. Such enzymes are called constitutive enzymes, and genes for these enzymes are constitutive genes or housekeeping genes. Next comes the luxury genes or non-constitutive genes. These genes remain inactive for most of the time in the lifespan and are expressed in certain cells or at certain time only when their products are needed. These are called specialist genes or luxury genes. The genes can be activated or induced to increase the level of mRNA and protein products. These genes are regulated by the action of RNA polymerase and molecular signals such as regulatory proteins, metabolites, or any other environmental cues. Next comes the structural genes. Number 3. These genes code for the substances which are needed for the morphological or functional traits of the cell and are called cistrons. This occurs in the split forms, segmented into introns and exons in eukaryotes, but in prokaryotes these are continuous. The structural genes are concerned with the synthesis of a polypeptide chain or a number of polypeptide chains and also with the synthesis of different types of RNAs which were required during the protein synthesis. The polypeptides thus synthesized may be used as component of plasma membranes or carrier proteins or as transport proteins or as an enzymes. Next comes the control genes. Some genes produce proteins that exercise a positive or negative control on the functioning of structural genes. These are of the following types. Number 1, regulatory genes. These genes actually code for the repressor or inhibitory proteins which regulate the transcription of other genes. Next, operator genes. These operator genes, they actually act as switch and turn on or off the transcription of a structural gene as and when required by the cell. This operator genes does not code any products. They are actually the sequences of DNA where the other regulatory proteins can bind and facilitate the transcription of their downstream genes. Next, the promoter genes. These genes provide sites for the binding of RNA polymerase enzymes for the transcription of RNAs by structural or luxury genes. And obviously, these promoter genes does not code any protein products. They are the sequence just for the binding of RNA polymerase. And last one is terminator genes. These DNA segments lie in the end of the message and stop or terminate the transcription activity of all types of genes. So, among the control genes, only regulatory genes produce some products, but rest of the genes, that is the operator genes or promoter genes or terminator genes, they will not produce or they will not code any message for protein products. They are just the sequences of DNA and facilitate the transcription or termination of the transcription. In prokaryotes, the genes and proteins are collinear. That is, the sequence of nucleotides in the gene corresponds exactly with the sequence of amino acids in the protein. Each gene is a continuous stretch of DNA whose length is related to the size of proteins it synthesizes. Thank you. Here the first section is ended. We will meet with another chapter under the microbial genetics in our next session.